Welcome to the University of Plymouth Skills in Biomedical Sciences. Today we're going to be doing the MTT assay. Now the MTT assay is based on the use of a thiazole tetrazolium salt which changes colour according to the metabolic activity of mitochondrial enzymes. So this MTT assay is used to measure metabolic activity and relate to cell death, cell proliferation and cell viability. Before we start, it is important to indicate the safety aspects of using MTT as part of this viability assay. Now MTT has been described in the materials safety data sheet for this chemical to be a skin irritant, an irritant to eyes, a respiratory irritant, and is suspected of causing genetic defects. So to start this assay, we are going to be using MTT as indicated. Now MTT is provided as a powder and we make it up at 5 milligrams per mil in PBS. We've already done that, we've filter sterilized it, and what we're going to be doing is adding it on to cells that are being cultured overnight. We are going to be adding it for, depending on the metabolic activity, between 2 to 4 hours, after which time the MTT should be reduced and we get insoluble purple formazan dye crystals, which we will solubilize later on in the assay. So by way of introduction, I've got an experiment whereby on a 96 well plate I have seeded 1 × 10⁵ cells per well, and I've treated them with certain biological agents that I want to test for cell viability. And what I'm going to do now is, after making up the MTT at 5 milligrams per mil, filter sterilizing, I'm now putting on 20 microliters of that MTT solution for every 200 microliters that are in the well of this 96 well plate. So I'm going to do that using the filter sterilized MTT, and this will take some time, and once done in each plate I can then culture in the incubator for 2 to 4 hours, after which we can then harvest the crystals. Okay.
A few moments later.
Okay, finally, after we've harvested the supernatants, we are left with the insoluble crystals. The final bit is to solubilize them, and what we do is we add 190 microliters of DMSO, and the DMSO will solubilize the crystals into a clear purple liquid, and we can measure that absorbance by the plate reader. So I'm going to add the DMSO to each well that has seen the MTT. And what can we clearly see is a nice purple colour that's been solubilized from those crystals. So finally guys, we have the plate, and this has been solubilized, and we've got a nice purple colour showing from solubilization of those formazan dye crystals. We're going to read the absorbance, we're going to put it into a plate spectrophotometer, and we're going to read it at a dual wavelength absorbance at 570 nanometers for the test and 620 nanometers for the reference. So we've got it in the machine. I'm just going to tell the machine, click on read, and replace the previous data set and it gets taken into the spectrophotometer. So in a couple of minutes, all the absorbances will be read for every well in the 96 well plate for both 570 and 620 nanometers. Okay, it's been a few seconds. The spectrophotometer has finished reading and it has put all the results up on the screen in front. I have saved this, and these are a collection of absorbances at 570 at the top of each well and 620 nanometers at the bottom of each well. Now what we've got to do is calculate according to the reference wavelength. So we subtract the bottom value from the top value. Once we've done that, we express all these calculated test data as a percentage of the control unstimulated or untreated sample, which is set at 100%. From that, all data can be converted to percentages relevant to that control and can show quite clearly the effects on viability, proliferation and cytotoxicity. I hope this has proved useful for you understanding the workings of the MTT assay.